Thank you so much for this kind introduction. I hope I can live up to the expectations. It's going to be a shift of gears. Let me just find my screen. Before I jump in, I'm interested in getting to know the audience. So who of you has ordered genetic testing? Okay, so, you know, a good number. I hope that I can teach everybody a little bit here with this talk. I've been actually part of this journey almost from the start, and it's been quite a ride. I've witnessed an explosion in knowledge, and I hope I can get some of the excitement over to you. My disclosure slide is here. So what will I cover? I don't need to tell you much about the background of inherited cardiomyopathies, but I have one slide. I want to focus on over a decade actually now, of clinical genetic testing and the lessons we've learned and what we still don't know. As with the prior talk, there is a lot of uncertainty in my field as well. And I really want to drive home the principle of clinical validity, which is becoming so much more important every day now that we can sequence and test so much. So the question is, what should we do, not what can we do? And if I have time in the end, I want to get into the future, from reaction to prevention, and ask the question whether we are ready for that. So with that said, these are the main inherited cardiomyopathies. I will not tell you what I normally say when I bring up that slide, because you know all that. The main ones, DCM, HCM, ARVC, all of them are collectively, the idiopathic cases are collectively very common actually. More than 1 in 500 individuals have a genetic cause. And as you well know, they can lead to sudden cardiac death. And on the right, you see a figure from Barry Maron's nice paper in 2007, showing in fact that HCM is the most common cause of sudden death in athletes. So now, quickly from your territory into mine, we know today that all of these cardiomyopathies have a substantial genetic component, much more than we thought, which creates a high incentive for predictive testing, especially for family members of an affected individual. If you're able to find the cause in the proband, you can now turn to testing the family members and potentially releasing them from screening. Quick breeze through the biology of this. The cardiomyopathy mutations collectively really affect a lot of different structures in the cell, ranging from the nucleus over the sarcomere to the cellular membrane and everything in between, really. But by far, the most commonly affected structure is the sarcomere, the contractile unit of the heart muscle. And really taking us back to high school biology, you see the blow-up of the sarcomere below, which has the thick filament in the middle and the thin filaments on top and below. So the actin, thin filament, and contraction is mediated by the myosin heavy chain gene, MYH7, binding to the actin filament, and the yellow pieces contracting, flipping over, and catalyzing muscle contraction. So I want to quickly get into the genetic testing. When and why do we test for genetic variants? Today, a lot of medicine and genetic testing is no difference, is that you start doing it when somebody is sick. So when we're treating symptomatic individuals, for genetics, this means you can either establish or confirm a clinical diagnosis. And as I said before, you can then, in the event that you really find a pathogenic mutation, do family testing, and those family members are the ones who benefit the most today, because you may be able to release them from screening, clinical screening, when they're negative. On occasion, in cardiomyopathies, not so common, there is treatment. So an example from our world is Fabry disease, which is a metabolic form of cardiomyopathy and can masquerade as isolated left ventricular hypertrophy. So you can mistake it as a clinician if the other symptoms are not yet or not obviously present. And if you now have a genetic mutation in the Fabry disease gene, you can treat it. That's great. But where we really want to get to is to test patients, or people, I should say, before the onset of symptoms, pre-symptomatically, and including those who don't have a family history, and then try to treat them before they develop symptoms to prevent disease if at all possible. I'll get back to that at the very end, because it's not as simple as it sounds. Here's a nice history of genetic testing. This is a relatively young discipline, actually. It started in the 2000s, really, and the upper part is from Barry Marin's nice review, showing you that the genetic testing really was kicked off by the discovery of the first HCM gene, MYH7, the one that I showed in the last slide, the thick filament gene. It took more than 10 years for the clinic to adopt clinical testing. And why was that? Well, because in those days, genetic testing was very complicated. It took us a lot of work to sequence just a single gene. And I don't know how much you know about genetic testing, but it is super easy today to sequence hundreds, thousands of genes, even the genome. So that's no longer the problem, but back then it was. So below, you see an expanded timeline for the last 10, 15 years. And now this is from the perspective of the Laboratory for Molecular Medicine, which is affiliated with Harvard, which is where I spent most of my last 10, 15 years developing this type of testing. We started in 2003 with a very small panel for hypertrophic cardiomyopathy. And over the years, 4 years later, expanded to add tests for the other cardiomyopathies one by one. In 2011, we're experiencing dramatic technological advances. You probably have heard of next generation sequencing that enabled us all of a sudden to test all these genes together. And then we and others in the community switched more and more to what's now very common, to just test multiple diseases at once. And I'll get into what the benefits of that are. So, quick look back. What have we learned over the last 10 years? We have learned that cardiomyopathies are genetically very heterogeneous. You see, I'm bringing them both up here, hypertrophic and dilated. These are data from the Laboratory for Molecular Medicine, showing you really that the detection, so that there's multiple genes involved for both, and the detection rates are sadly still very modest, despite the fact that we've been doing a lot of discovery. 30 to 40% at best in an unselected referral population. Now, if you test people with family history, you will have higher detection rates, but they're not great. Hypertrophic cardiomyopathy is genetically simpler. Just 2 genes, as you can see, make up most of the positive cases. That dilated cardiomyopathy is much more complex, in a sense that there are more genes, and there's only one gene that stands out a little bit, but not quite as much as for hypertrophic. This is the titin gene, the largest protein in the genome. No other gene there really makes up more than 5%. So lots of genes to test. Not only have we learned that there are many genes, but there are also many different variants out there in the population, and the vast majority still, to this day, continue to be unique. That means you find them in one patient, in the one you're testing, and never again. So now, you as a physician may not care about that, but from a laboratory perspective, this is very annoying because we have to continue to sequence all these genes to find all these unique variants, and that makes for expensive tests. So, in summary, 2/3 of these variants that we found over the last 10 years were seen in only one patient. And then it tapers off quickly. And there's very few, just a handful of cardiomyopathy variants that you see relatively frequently. But this isn't good enough to make a very focused, small, inexpensive test. So as I said, you have to continue to sequence. And we also all underestimated clinical heterogeneity. That makes for diagnostic odysseys, as illustrated by this case that I brought that came onto my desk repeatedly, actually. So there was a proband with a clinical diagnosis and a family history of dilated cardiomyopathy. This is denoted by the arrow in this family tree here. And we ran, as was customary at the time, a DCM panel and found a variant, but that didn't really segregate with disease, and so it was inconclusive, nothing that we could tell the family. A year later, the diagnosis was revised to arrhythmogenic right ventricular cardiomyopathy, which is not uncommon. We then ran the ARVC panel, and that was positive, and that variant really was present in all affected individuals we were able to test, meaning that we very likely found the cause of disease. We know today that this is not uncommon. It's not the norm, but it's not uncommon by any means. About 3% of dilated cardiomyopathy patients have a variant in an ARVC gene, speaking to this overlap between the disorders. So underscoring why traditional testing really makes no sense here. Just do one test and then the other. And that's another way of looking at it. This multi-disease testing paradigm that we're now using, this is the tip of the iceberg analogy. In the old days, clinical diagnoses were defined on by morphological criteria, meaning that we naturally focused on the most severe cases to define our diseases. And as laboratories, we would then make diligently one test for one disease and so on and so forth. And as I mentioned before, we replaced that with this to accommodate for the fact that under the surface there is a lot more unrecognized clinical heterogeneity and overlap between these disorders. So sequencing is becoming cheaper and cheaper, and what that has caused is an explosion in knowledge, a lot of discovery being done. And what you see here is a graph showing the number of published disease genes that have come out since 1990, when MYH7, the first HCM gene, was discovered. So look at this today. In 2014, now this is actually not today, this is 4 years ago, we were already at over 50 HCM genes and over 60 DCM genes, and the counts are rising. Now, question is, are these all real? We didn't really ask that question for a long time. We just kept adding genes to our panels, and I'm guilty of that. You know, this was an exciting time, and we tended to believe the literature when it said this is a disease gene. So what this led to, is a realization that these ever-growing gene panels can be Trojan horses. What you see here is a graph showing the detection rate over time for dilated cardiomyopathy in our laboratory from 2007, where we did 5 genes, and then in 2011, when it jumped to 46. Now, yes, we quadrupled our detection rate, which was great, so going from 10% to almost 40%. But look at the black line. The number of inconclusive cases rose even more, so that is no good. So that begs the question, is more really better? We are still firmly rooted in the panel testing world, meaning, you know, if an order will come in to test a patient for, let's say, HCM, and we do an HCM test, now we may have a larger panel in the background. If somebody wants to do multi-gene testing, that's fine, but we are moving toward exome sequencing, if not genome sequencing, very quickly as a first-line test. So we need to be really critical and ask which genes should be on a panel, or if we do exome and genome sequencing, which genes should we look at? We can sequence them all, but clearly this may not be great. And here's another look at the HCM situation from a laboratory perspective. I plotted all the different laboratories that, 14 laboratories, there are more, offering gene panels, and the height of the bar shows you how many genes they have on their panel, and the colors show different subsets. Blue, yellow, green are ones that we've known about for a long time. They're relatively valid. Everybody across the board is testing for the obvious ones, so that's good. But look at the variability in the red. These are like all sorts of disease genes that, you know, we've all been adding, and no consistency whatsoever. So as a physician ordering this test, which one should you test? You know, what's your metric to evaluate these? The number of genes? Clearly, as I showed you before, that can have caveats, so that may not be the best. Which is why a few years ago, the US National Institutes for Health have founded the Clinical Genome Resource, or ClinGen, which is a large consortium that's focused on building standards for assessing clinical validity for a variety of questions. One is this gene associated with disease? That's the one that I want to focus on today. Is the variant causative? Is a whole other question that is very important. And then lastly, is it actionable? We do that I'm part of the Clinical Genome Resource. We have clinical domain working groups, and a subgroup of us, have focused on this question: which gene is really reliably disease associated for HCM for the last few years. You see the members here, and my co-chair Ray Hirschberger. So this work has just been completed, and first author Jody Ingalls is in preparation. Hopefully you'll see that in press sometime soon. It's sobering. This graph shows you all the published HCM genes, so over 50. And the bottom line is the evidence for many is not great, actually, if you look critically. The height of the bar here denotes the strength of the evidence measured by various means. And you may not be able to see the details, but what you should focus on is the entire right side of the graph shows the genes that have either limited or no evidence, so the majority. A few, I think 3, have moderate levels of evidence, and only 9 of all of these were deemed to have definitive or strong evidence. And what's even more sobering in my mind is that there's only one single gene that we haven't already had on our tests for the last 10 years. So the pendulum is swinging back quite a bit. You know, we have all these proclaimed disease genes. Now, it may very well be that they are, but we just don't have the data yet to show that conclusively. So what we need to do is really move toward evidence-based test content selection. So the more is better, it didn't work out in my mind. But we have no guidance. Currently, laboratories choose their content on their own, and some do a good job, some do a bad job. You don't know as a provider unless you do the work yourself. So we need to make decisions as to what level of evidence qualifies a gene to be included in what type of test. There's diagnostic testing, and I think currently most laboratories include moderately supported genes in their diagnostic panels. Predictive tests, everybody would agree you need more evidence, so strong or definitive. But really what we need is professional society guidance here, and I would personally argue that we also need regulatory enforcement, forcing laboratories to make clinically valid tests. I'm almost at the end. Going back to this question, when and why to test for genetic variants. So we covered the diagnostic piece, and it's incredibly tempting to think about testing pre-symptomatically everybody, not restricted to individuals with a family history. So that would be great, but we're learning quickly now, as we're starting to do this, that it's not as simple as that. I don't have enough time in this talk. This would be a whole different talk, but I want to leave you with these 2 papers that are really, really incredibly interesting to read. We're starting to sequence healthy individuals, and we're learning that many of these pathogenic variants that we thought we understand so well just don't seem to cause disease in as many people as we thought. So meaning that we have been focusing on a population of people with disease, and we understand the variants in those individuals, but they also have, everything, they have the, particular backgrounds in their families, probably predisposing them to disease through additional factors. So now we're testing these genes in healthy and find that it's not quite as simple as we thought. So I personally think we have quite a way to move to truly predictive screening, but it is coming. And with that, I'd like to summarize. Inherited cardiomyopathies have a strong genetic etiology. The outcomes are potentially severe, and we have a strong incentive for genetic testing. Currently, this is done mostly for pre-symptomatic individuals, but we're moving toward predictive testing in everybody. We have clinical and genetic heterogeneity, leading us to build these multi-disease panel. We're quickly moving toward exome, if not genome sequencing in the near-term future. More is not better. This is my main message to you. If you order tests, don't assume the ones that have the most genes are the best. Clinical genetic testing panels are not standardized. You really need expert guidance. And we just talked about the predictive testing is starting to be discussed, but insufficient understanding of the penetrance of our variants will, make this, we'll make it take a little bit longer. So with that, thank you very much for your attention.